Hello, my name is Robert Bone, and I am one of the authors of this manuscript, which describes how altering metabolism programs cell identity via NAD-dependent deacetylation. During mammalian preimplantation development, at around 3.5 days after fertilization, the embryo consists of two lineages, the trophoblast and the inner cell mass. Roughly 24 hours later, the embryo now consists of three lineages, the trophoblast, and the inner cell mass has split into the epiblast, which will form the embryo proper, and the primitive endoderm, which will generate the yolk sac. As development progresses, the lineage factors that were co-expressed in the 3.5-day-old inner cell mass become spatially segregated into separate lineages that have more restricted developmental capacities. In the Brickman lab, we routinely use embryonic stem cells, or ES cells, for our experiments. These are cells which are derived from embryos at the peri-implantation stage, as shown here, and consists of a heterogeneous mixture of cells, primed towards either the epiblast or the primitive endoderm. We maintain these populations in conventional stem cell culture media, referred to here as serum lymph. For several years, the Brickman lab had known that there were conditions in which small populations of cells which co-express these different lineage factors also existed. When we asked what was the common feature of these conditions, we found that it was alterations in cellular metabolism. In this manuscript, we force cells to change their metabolism by exchanging components of the media to make enhanced metabolic media, or EMM, where cells appear reprogrammed to an earlier ICM-like state, where cells exhibit enhanced self-renewal and differentiation competence. We think of these as super ES cells. We tested the ability of EMM to enhance ES cell behavior, or generate EMM-cultured ES cells, or EMES cells, by injecting them into a preimplantation embryo, then transferring the embryo into a pseudo-pregnant mouse, and analyzing the contribution of the EMES cells to the host embryo. While serum lymph-cultured ES cells and another commonly used culture, 2I lymph, contribute modestly well to the epiblast, EMES cells contribute exceedingly well to the epiblast, and even show some contribution to the extra embryonic lineages as well. We next investigated how EMM brings the enhanced fitness or superstem cell phenotype. Normally, ES cells are cultured in glucose-rich media, in which glucose is transported into the cell cytoplasm, where it enters the glycolytic pathway, generating 2 molecules of pyruvate for every 1 molecule of glucose, which is then either processed into lactate or transported into the mitochondria to be processed in the TCA cycle. However, in EMM, glucose is replaced by its stereoisomer galactose, which inhibits the first enzyme of the glycolytic pathway. This blocks sugar metabolism entirely and forces the cells to metabolize other energy sources, namely fatty acids in the mitochondria. Meanwhile, in the nucleus, relatively high levels of acetylation of histones and transcription factors results in many open enhancers with low levels of transcription for many genes that are not essential for lineage specificity. We notice that acetylated transcription factors in this context cooperatively bind to open enhancers, but not very strongly. However, in EMM, the NAD-dependent deacetylase Sirtuin is activated and acts to selectively deacetylate both histones and transcription factors. This results in many non-specific enhancers being deactivated, but because the transcription factors are also deacetylated, they continue to bind to the highest affinity enhancers. This select set of lineage-specific enhancers are still acetylated. At these open enhancers, we detected enhanced cooperative binding of key transcription factors. The overall effect of this is that we see the dampening of extraneous enhancers and increased signal from those that are lineage relevant, which generates an increased transcription and epigenetic signal-to-noise ratio that could be critical for maintaining stem cell fitness and longevity.